In labeled line coding, an individual sensory receptor is selective for specific stimulus characteristics and communicates to neurons in the central nervous system that are selective for the same characteristics. For example, a spiral ganglion afferent is selective for specific frequencies of sound and communicates to neurons in the CNS that are similarly selective. In population coding, stimulus characteristics are coded by a distributed pattern of neural activity and not the activity of any one neuron. For example, the pattern of activity across all glomeruli in the olfactory bulb can be used to decode the identity of an odor.
So how are odors encoded when we get up to the bulb? One way this has been addressed is using calcium imaging of neurons in the olfactory bulb. This data was collected using GCAMP, our genetically encoded calcium indicator protein. Here it was expressed in cells in the olfactory bulb. And this allows us to look at the change in activity when different odorants are applied to the nose. Here we're imaging over a big swath of the olfactory bulb, and in color here we can see the size of the response. The more red the color, the stronger the response, and the more blue, the weaker. These blobs that you see appearing here, these are actually individual glomeruli, where you have the axons from the two types of neurons coming together. What's seen generally when you apply odorants and collect these images is that individual odorants activate a broad swath of the glomeruli. And that should be expected based on what I told you before, that an individual odorant can activate many different receptor types. And all those receptor types send axons up into the bulb to different glomeruli. So we get sort of a broad distribution of activated cells. So this is an example of what we call population coding, where stimuli are coded by different levels or patterns of activity across a population. The idea here is that you have to look at the whole population to know what the stimulus was, because many cells are responding to many different stimuli. Their responses aren't specific to one stimulus. What is specific to a stimulus are the levels of activity or the patterns of activity across the population. For example, we have a strong responding cell here, a weaker responding cell over here, some weak responses elsewhere, and together that makes up the representation. So it's not just what cells are active, but the patterns of activity or the levels of activity across the whole population. That's really the essence of a population code. An important corollary here is that you can't just identify the stimulus by looking at which cells are active. Again, because cells are going to be active for many different stimuli. But we'll see later in the lecture that this is actually very different from what's going on in the taste system.
In introductory classes, you learned about labeled line and population coding. But neuroscience is an interdisciplinary field, so there are many words for these terms. For example, labeled line coding may also be called the labeled line principle, or labeled line theory, or labeled line hypothesis. Similarly, population coding may also be called combinatorial coding. Both labeled line coding and population coding fall within the umbrella category of neural coding, which includes many theories about how the nervous system represents sensory information.
Generating tools that can decipher population coding is an active area of research. While patterns of activity may vary from one individual to another, scientists are beginning to develop algorithms that can decode sensory characteristics from patterns of neural activity. In contrast, labeled line coding in the early visual and somatosensory systems is predictably consistent across individuals, and knowledge of these pathways plays an important role in clinical neuroscience. If a patient complained of an abnormal sense of vision or touch, the nature of the abnormality could be used to precisely pinpoint the likely location of an injury that is causing the sensory abnormality. For example, if a patient complains of a dark spot in the upper right visual field, knowledge of the labeled line pathway for vision could be used to narrow down the potential location of injury to a small number of subregions in either the eye, the optic nerve, the optic tract, the thalamus, the cortical white matter, or even the primary visual cortex. The location could be further narrowed down by determining whether the visual abnormality disappears when closing one eye or the other.
Now that you've had a bit of review on labeled line coding and population coding, let's see if you can follow along with this example of labeled line coding in the somatosensory system. In this example, we'll start with a nociceptor that is located in the hand. This nociceptor would project towards the spinal cord in a spinal nerve and have a cell body in the dorsal root ganglion. From here, if we were dealing with a nociceptor in the hand, it would project to the cervical part of the spinal cord, where it would synapse in the dorsal horn. From here, the second neuron in our pathway is going to cross over to the opposite side of the spinal cord and ascend up to the thalamus. The third neuron in our pathway will project from the thalamus to the primary somatosensory cortex. Okay, so at each step along the way here, we would have neurons that are selective for noxious stimulation of the hand. The nociceptor, when it fires, would signal that there has been injury to the hand. This second neuron in the spinal cord, which the nociceptor synapses with, when this neuron is firing, it too would indicate injury to the hand. And similarly, this thalamocortical neuron, when it is firing, would indicate injury to the hand. So we can see that this is a labeled line pathway because activity in any one of these individual neurons is going to indicate stimulation of the hand. By contrast, if we were thinking about a population coding, the activity in individual neurons would not have identified the stimulus, but rather we would need to look at the pattern of activity across many.